Hello students, welcome to my channel Online Biology Tutorials. We are going through important questions of chapter number 11 and this is video 2 of the important questions of chapter number 11. If you haven't watched video 1 for chapter 11, please do so. Okay. Now here again I have picked few MCQ questions which you can go through because obviously you can use them while answering your answers in the exam, because you must be aware in term 2 marker, 3 marker and a 5 marker question will come. Now question number 1 is asking lysozymes kill bacteria by destroying. So of course they act on the cell wall. The group of letters that form same words when read both forward and backward direction, this is palindrome. So if a question comes on palindrome, you may write the group of letters that form same words when read both forward and backward. So this is the definition to palindrome. Next question is the techniques of using live organisms of enzymes from organism to produce products and processes useful to human beings is of course biotechnology that we have studied in chapter number 11. Kanamycin does not inhibit the growth of. So kanamycin will not be effective in the case of E. coli because otherwise E. coli does not carry any gene of antibiotic resistance. Next question is again on PCR and it is asking us that to produce copies in millions of our DNA segment, the number of times PCR should be done. It is 28 to 32 times because this is the highest value. If you will think logically, this will yield maximum number of copies and it would be somewhere in billions, because in the previous video also we have read that if you want to calculate the number of copies, the cycles will be given to you. So here 2 to the power number of cycles will give you the answer. So here it is giving us the maximum value that if 2 to the power would be 28 or 29, 30, 31, 32, this is going to yield maximum results out of all the given options A, C and D. This has been explained in video 1 also. So if you want to have a look, you can go to that video to check it out. Question number 6 is asking a recombinant vector with the gene of interest inserted with the gene of alpha galactosidase enzyme is introduced into a bacterium. What is this method of selection referred to as insertional inactivation? So for this I'll be sharing a link in the I button. You just go through that video because this is 100% theoretical based question. Next question is, can you recall meiosis and indicate at what stage a recombinant DNA is being made? So if you recollect, then recombinant DNA was being made during the Pachytene stage of meiosis 1. Okay, Pachytene stage of meiosis 1 and prophase 1. You know, CBSE is going out of the box these days. The portion that was being deleted in chemistry, it was asked, the isotherm one was asked in the exam. So I would like to suggest everyone that please go one level up so that you can get good marks, right? So meiosis stage is being defined as a Pachytene in which recombination occurred. Which was the first type 2 restriction endonuclease to be discovered? So it was none other than hint 2. What is Plasmid? So Plasmid is tumor inducing plasmid of Agrobacterium tumefaciens, and this plasmid is being used as a vector. You can also say that plasmid was used during the RNA interference technology to make tobacco plant resistant to a nematode Melidogyne in Cognitia. Please try to introduce all the terms that are related to the chapter and not in general. Next question is, explain the role of alkaline phosphatase in recombinant DNA technology and what is the source of this enzyme. This question was asked in a different way in video 1. There also I took this question. Here again I am taking it. So alkaline phosphatase is a one when you treat restriction endonuclease with your DNA segment that you have isolated because you want to extract gene of interest. So once you treat the DNA that you want to work upon with restriction endonucleases, you'll get sticky end. Similarly, you will treat the plasmid DNA, you will also get the sticky end. So until you don't want both of them to combine and form recombinant DNA, this enzyme is being used, which prevents the self-ligation of the sticky ends of your gene of interest and sticky ends of the plasmid DNA. So it has two origins. It can either be taken from bacteria. If it is taken from bacteria, it is known as bacterial alkaline phosphatase. I'm not writing in full, so it will become BAP. And it can also be taken from calf alkaline phosphatase. So if it is taken from calf, it will be abbreviated as CAP, right? So alkaline phosphatase is one of the important enzyme which prevents the self-ligation of two different types of DNA. Now enumerate the steps. This question has also been taken in video 1, where we talked about that first isolation of the DNA will be done, then treatment with restriction endonuclease will be done, then isolation of the fragments will be done, then what will be done? We will ligate the DNA to form the recombinant DNA, then it will be introduced in the host, and then ultimately the host will be cultured, right? So the steps are written in video 1. If you want to go through the steps, you can go through video 1 again. Thank you. Now next question is question number 12. Can you think and answer how a reporter gene can be used to monitor transformation of host cells by foreign DNA in addition to the selectable marker? Remember, whenever they are talking about reporter gene, then here they are talking about the insertional inactivation process. You know, suppose a plasmid is there and it carries a gene, and this gene will produce the enzyme beta galactosidase. Now this is the same enzyme which we have read in the lacoperon, and this enzyme help in breaking lactose to glucose and galactose. So if you introduce your gene of interest here in the beta galactosidase region, then it is not going to produce beta galactosidase, and of course the lactose will not be broken down. Here the lactose was made up of a special component that was glucose plus X-gal, and this X-gal was chromogenic in nature, means whenever it would be produced, it will produce a color. So if your gene of interest, if your recombinant DNA has been inserted into the beta galactosidase gene, it will become inactive. No chromogenic substrate will be formed. So this is insertional inactivation. For this also I'll be sharing a link in the I button. You please check out from there. Okay. Describe briefly the downstream processing. So downstream processing is the last step which is being mentioned in chapter number 11. Separation and purification is the keyword. Separation and purification of the proteins that are being collected from bioreactor is done. Then a preservative is being added. Then the material goes to the quality control department of an organization, where if it passes the quality check, then it is being released and commercialized in the market. Okay, so I hope you have understood all the questions. I have few more videos on this chapter, so stay connected and do share it with your friends. Thank you.